हेलो। 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 सर, कैन यू हियर मी? हेलो।
शायद ज्वाइन
ज्वाइन तो किया हुआ है, मिलेगा के नाम से पढ़ा तो है। ये वे सर जब आएंगे, सर का पीपीटी भी तो लेके आना था। श्रुति, सर का पीपीटी लेके आना था। मृदुल से कह दो सर से पीपीटी लेके आ जाएगा पीडी में। पीपीटी तो लगाएंगे ना। या तो आप लेके चले जाओ, जैसे भी है। जैसे जब तक मैं बोलूंगी ना तब तक पीपीटी सेट कर देखिए सेट कर। अरे ये तो आपका है, सॉरी। मेरा कहाँ गया? मेरा उधर है। मेरा
A very good morning, everyone. On behalf of Amity University, Rajasthan, I extend a very warm welcome to all the dignitaries, faculty members, participants, and students who have joined us here for the conference on sustainable development in applied science, engineering, and technology SDSET 2025. We are truly honored to have distinguished keynote speakers with us today. Professor Vinay Sharma, Dean Research Director, Amity Institute of Biotechnology, Amity University, Rajasthan. Dr. Madhusudanan UK, Additional Professor, Department of Biochemistry, Sri Chitra Tirunal Institute of Medical Science and Technology, Trivandrum, Kerala. Dr. Ragini Singh, Additional Professor, Department of Biochemistry, All India Institute of Medical Sciences, Rajkot. So, to begin today's session, I would like to invite Dr. Vijay Chaudhary Ma'am, Professor, Amity Institute of Biotechnology, Amity University, Rajasthan, to kindly deliver the welcome address. So, please welcome ma'am with a huge round of applause.
Very good morning to everyone. I hope all of you had your breakfast and is full of energy. Welcome back to the third day of International Conference ICGETI 2025. I would like to take the privilege of welcoming our dearest Professor Vinay Sharma sir, Dean Research, Amity University, Rajasthan, and Director, Amity Institute of Biotechnology. Sir has over 36 years of teaching and research in plant science. Earlier, Sir served as Associate Professor at IIT Roorkee and Dean and Head at Banasthali Vidyapeeth. Sir has major achievements, research achievements, a total of 443 publications, 393 in research journals, 50 conference proceedings and book chapters, 4 textbooks, 5 edited books, and guest editor of 5 research journals on thematic issues. Sir's bibliometrics metrics include an H-index of 47, I-10 index of 181, and over 8,358 citations. Okay, sir. Okay, sir.
From this place.
Okay, sir. The PPT note can let us
All right.
Sir has research interests in plant stress biology, plant biotechnology, plant informatics, lignocellulosic bioethanol, secondary metabolites, in-field photosynthetic performance. Sir has guided 77 PhD, which has already been awarded. Many of them are well-placed and employed as faculty in Indian and foreign universities and industries. Currently, Sir is mentoring several young scientists and research scholars for innovative research in plant science at Amity University, Rajasthan. And Sir has successfully handled a lot of research and development projects funded by DST, ICAR, DST-DASH, DST, JSPS, DPT, UGC, DRDO, and state DST supported major projects as PI and co-PI. Besides, Sir have been the coordinator of DST FIST, DBT Center for Bioinformatics, DBT MSc Biotechnology programs. Sir is life member of 9 prestigious scientific bodies. Sir has been awarded with a lot of medals and recognitions. Sir, research contributions have been recognized by the following prestigious fellowships and awards. Fellow of National Academy of Sciences, Fellow of Biotech Research Society, Fellow of Botanical Society, and many more. I request Sir to start the presentation so that we can continue. Thank you, Sir. Thank you for coming.
Okay. So, all right. Thanks a lot for this wonderful session and introduction. I hope I am audible to all of you. I doubt. It working. Am I audible?
Yes.
Now. See, now I am. Absolutely. So actually, how much time we have now? Dr. Vijay?
Sir, we have half an hour, 45 minutes. Half
Half an hour or 45 minutes?
an hour. Half an hour.
Half an hour to 45 minutes.
Sir,
अरे अभी तो आप 1 घंटा 45 मिनट लगेंगे
आपके ऑफिस जाके आई
काफी स्टार्ट कर रहे हैं
अच्छा वो बस फेलिसिटेट करना था
Yes,
सर को
I would like to, invite our conference chair, Professor Manju Kaushik ma'am, and I will request ma'am to kindly felicitate sir. अपना कॉन्फ्रेंस कहाँ?
Background
ICGETEI. इसमें था।
ये रही एक
देखिए। इसको अनशेयर करके करेंगे। अनशेयर करके करना।
ये यहाँ से स्टॉप शेयर हाँ good, yeah. शेयर
शेयर।
I'm sure it's working. एक बार ये पीपीटी हटा एक बार पीपीटी क्लोज
शेयर ही नहीं होता है क्या?
स्टॉप शेयर पीपीटी यहाँ से जो है ना यहाँ ये क्लोज करो
अब आ जाएगा।
जी
So well, here again. So in fact, I know that we have little time, and my idea is not to focus on very intense research. My whole idea is to expose you to what we have done, what we are doing presently, that's one. But not going into the nitty-gritties of that, that's one. Second, I mean, I find that most of your undergraduate students and then some of your postgraduate students also. So you will be wondering that, how this intensive research lecture, which of course I am not taking research lecture, is going to help us. Now I must share with you that in our country, we have some, over 5 lakh startups, 5,50,000 or so startups are here. And out of these startups, I wish to emphasize again, some 150,000 startups have come from the undergraduate students. So think about it. That means your contribution is approximately 25% of the entire contribution. You don't have to, go in for high five, very kind of in-depth studies and then be PhD, be all those things. I mean, they are necessary, of course. That is where you will again get a lot of ideas there also. But as an undergraduate as well, you can, do a lot of things. Don't think that we cannot do. I repeat again, 25% of the startups in India are coming from undergraduate like you. So have that kind of mindset, cultivate that kind of mindset. Be, observant and be try to catch what are the problems around us and then try to find a solution to all those problems. So I would talk about a very important area of research in plant sciences. As for instance, we have stress, we have infections, we take antibiotics and so on, and then we are also prone to different kinds of environmental fluctuations, Isn't it? We feel thirsty, and if we don't get water, and if temperature is high, etc., we react. So put together, we have a similar kind of stress in plant stress. Plants are also prone to infection by pathogens, different kinds of fungi and bacteria are there. We have that category of stress is called as the biotic stress put together. Biotic, because coming from living organisms, all types, even viruses. So we have viral, bacterial, fungal, and so on, and abiotic. That is, for instance, we have drought, we have in the field the crop is growing, and we have drought, we have high temperature, we have, let's say, deficiency of mineral ions, because minerals are required for the proper growth of the plants. So all those things happen. So we have abiotic and biotic stresses, and essentially these are responsible for huge losses to the crop. I mean, one way of, let's say, addressing the problem of food security is that you develop high yielding crop varieties, which of course within the same acreage can give you high yield in terms of the productivity. That is, they are more productive. That's what we are doing. The second way, and very potent, very powerful way of doing it, is to develop the plants which are resilient to stress, different kind of stress situations, because there again there are huge losses. Put together approximately crops losses, we combine biotic and abiotic stresses put together, crops have losses as high as 90% due to one reason or the other. Different, so all those things are happening. So if you do that, then again you are solving that problem. So another way of addressing, therefore, I repeat, of this food security is to develop these stress resilient crop. I do not really want to go into the details of all that, but population is also very you are aware, and most of the population growth is coming from the developing countries like India. India is now one of the fastest growing, despite the fact that we have slowed down our population growth. Nevertheless, we are still one of the fastest countries which are contributing to the overall world growth population. So it's an immense problem for developing countries. They have to really address this problem. So they have to all the time, be prepared to accept the challenges of food security and keep on working. So that's about the problem. Now, how we are able to address this problem scientifically? So again, WHO, half of the world population will be living in water-stressed area by 2030. So we are going to, this problem is going to be becoming more and more severe in future. So there are two ways to do it. Scientists world over are working in order to develop stress-resilient crops. One way to do it, that you bring the crops to the laboratories, provide them, grow them under different kind of stress situations. That's one way of doing it. And find out what is happening in the plant, so that once you know the mechanism, you can develop more robust crops. The other way to catch hold on of doing it, in fact, that is one approach we applied, and that was a novel approach of catching the plant which are growing naturally in the environment, in an environment which is challenged by different kind of stresses. Different kind of, especially abiotic stresses. So we followed that approach also. So naturally growing plants or bringing the plant to the laboratory, providing stress, and then studying different mechanisms. So, okay, so this goes on. We found several clues that how the plants are becoming more resilient in the natural environment. How the plant, one plant dies, the other plant is able to survive. So what are the mechanisms thereof? And we found several mechanisms. For instance, they are better mineral ion acquisition efficiency, same amount of phosphorus in soil. Two plants, one is stress resilient, is able to accumulate more. It has a better acquisition efficiency. It works under severe mineral ion nutrient deficient conditions. That's one. It has certain enzymes which are structurally modified. Their catalytic subunits are modified. For instance, we worked on the ATPase, vascular tonoblast ATPase. So that's a wonderful enzyme, and it gets changed. It undergoes changes in terms of subunits and structure when it is challenged with stress. Another aspect, they have a better long-time water usage efficiency. This we studied by using radioisotopic oxygen and carbon dioxide, and we found that the plants which are surviving naturally, they have a long-term water usage efficiency. That is, they are able to work and conserve water more than the plants which are susceptible to stress. So these were some of the mechanisms which we unfolded by that. But then, in nature, because I told so many different kinds of stresses, plants are often exposed to more than one stress. For instance, if there is a drought stress, then what is the guarantee that fungi will not come and infect the plant? So it becomes a combined stress. Or if the drought is there, where is the guarantee temperature will not rise? Or if the drought is there, where is the guarantee that the plant is growing under saline soil? So one is therefore in nature, the point that I'm trying to emphasize is that in nature, plants are challenged with different kind of combinations of stresses. Abiotic, combined, abiotic and abiotic combined, etc., etc. So all possibilities exist. This, for instance, is a deadly pathogen that will form in Fasciolina. It's notorious because notorious I have written that because it is able to, it's very widespread and it is able to infect both seed borne and soil borne, and therefore it is able to widely infect a large number of crops. So very deadly, and we lose a lot of there are huge crop losses because of all this. So we worked on this, what is happening. We found that certain enzymes like beta-1,3-glucanase is the one which is responsible for cell wall degradation. Chitinase is another enzyme which is responsible for fungal wall degradation. So they act jointly, and therefore they weaken the attack by the pathogen. That's what is happening. Or alternatively, their gene expression, etc., of these enzymes, that is also upregulated when they are challenged by the pathogen. So that's one study which we conducted in isolation only for the biotic stress. Another element which has come up now, we are having climatic changes. So we have biotic stress, we have abiotic stress, and now we are also challenged with the changes in the climate, erratic patterns. So all of you, I don't really have to address that. So three possible threats are there: climatic, biotic, and abiotic, and therefore we need to work on not just one. If we make the plant secure, if we make the crop secure by way of imparting the abiotic stress resistant genes. For instance, if it becomes drought resistant, where is the guarantee it won't be attacked by the pathogen? So you have to look and think in terms of developing the multiple stress resilient crops. So combined abiotic or abiotic and biotic, there could be multiple possibilities. And the whole idea is that, to identify certain factors. I mean, it's a complicated biochemistry and molecular biology, but I am saying put together as factors which are triggered by abiotic stress. you identify certain factors which are triggered by way of fungal pathogen attack, that is by biotic stress. Find out how many of them are shared and how many of them are unique. And you find that there are many factors which are common to both abiotic as well as biotic stress. So these become shared. So you have shared, you have unique, and once you identify all that, then you can develop, the multiple stress resilient crops by working on those factors which are the shared factors, which are the common factors. So we followed this approach, and one approach was, for different abiotic stress combinations, that we have salt stress, we have drought stress, we identified the proteins, and then let's say we identified 6 proteins. You were working on sorghum, which is again a very important leguminous crop, and we identified 6 proteins. Of them, 4 were common in salt and drought stress. And this we did by using the combination of wet laboratory and the bioinformatics approaches in silico approaches. So that's the way, because these days, you have to combine different approaches. Many approaches have to put together in order to build up a story, complete a story, and come at certain worthwhile conclusions. So, of course, all this is published as high as 10.2 impact factors. So, the second point, which elaborating on that, is combined abiotic and biotic stresses. So, what that story, macroforming of testosterone I just mentioned, in India, this accounts for 10 to 44% of reduction in yield. So that's a huge amount, and the pathogen is very deadly. I've already told you, I used to call it as an notorious pathogen. And it's not just in India, it has a worldwide distribution, all over countries, including USA, for instance, even Europe, South American continent, Africa, and so on. So it's a global menace, in fact. So we worked on Ligna radiata, which is mung bean. All of you are aware of this crop. It's one of the most common pulses, and it's rich and it's popular for many different reasons. Nutrition, nutrients, and proteins, and so on and so forth. So we worked on this relationship, macrofauna and mung bean, and we came out with a kind of, I would say, a novel of experimental approach. Just half a minute to explain that novel experimental approach. That not one is that you directly challenge the plant with drought and fungi together. That is abiotic and biotic. And second is that you have them independent. And then different combinations. That first abiotic followed by biotic. That is one combination. Abiotic, biotic, and then abiotic again, or biotic, abiotic, and then biotic again. I mean, different combinations in different orders. You have different orders, not that a set order, but different orders. And we very interestingly found that plants behave differently if you change that order, the order of the stress which you create in the plant. So, so I would not really dwell into this, because my whole idea is to conceptualize you, to give you certain ideas so that you are able to understand. So detailed protocols, I'll not go into the data of all this in detail. So you see, there are 2 possibilities, biotic and abiotic. One possibility is biotic and abiotic can work in synergistic way. Antagonistic and synergistic, or you are all there. They work in synergistic way, which means that if they are working in synergistic way, then they become very deadly. Because both are cooperating and both are challenging their plants. So it becomes very deadly. But if they are working in antagonistic way, that is, one counters the other, then the plant still is happily growing and surviving. and performing better than even when it was not really challenged with any kind of stress. Now that's again a normal contribution. I mean, that means you can develop strategies where you expose the plant to different combinations and increase. It will lead to increase in the growth of the plant, in the quality of the plant. So all this, we of course carried out the study in detail, light microscopy, to see that how the fungi is penetrating into the plant and how it is developing into mycelium and microexploration. And also did the FECM for this, again on the right side is the one which is exposed to the stress, and we find that the rounded global globe kind of structure that you find on the surface is that of the microexploration. And then the study went on to study different growth parameters, starting from elementary to advanced parameters like growth parameters, or the number of seeds, or the rate of seeds, all these parameters are important, or the seed protein, or the seed total antioxidant capacity, a lot of parameters were investigated, and a lot of biochemical parameters as well, because when you challenge the plant with stress, it reacts in different ways. And in how it reacts, there is oxidative stress, antioxidant system activation is there. So we have a whole lot of enzymes, whole lot of antioxidants. That is, we have both metabolites and enzymes which are produced under the stress conditions. So we studied some of them which are written here. For instance, glutathione, glutathione peroxidase, superoxide dismutase, and folic acid. So many of the parameters which I mentioned here, they were actually investigated by us. Another important thing, we have in animals, we have signaling molecules. You are aware. So plant hormones, the hormones are signaling molecules. In plants as well, we have signaling molecules, and the prominent amongst those signaling molecules being salicylic acid or jasmonic acid. Now, these are signaling molecules in plant, which, first of all, are produced as there is a attack by the pathogen or under abiotic stress conditions. So these signaling molecules, that is the first event, and then they lead to a trigger of reactions by the plant, which would lead to overcoming that kind of stress. So that is what happens. So we have salicylic acid, which is again important for regulation of the antioxidant system. That is one. And second, we have, when the plants are challenged with pathogen, we have a whole lot of proteins which are called as PRPs, pathogenesis related proteins. So under that, they are also governed by signaling molecule like salicylic acid. So we studied, in fact, both of them. So a lot of biochemical parameters like chlorine, proline, cat enzyme, GPX, SOD, hydrogen peroxide, malondialdehyde. So all these are antioxidants, and also the photosynthetic parameters by which you can perform using the equipment like dual band, where you can assess the photosynthetic efficiency of the plant when it is growing in natural conditions. So you can have a real-time testing that how it is performing in the field, and you are able to get the idea of both PS1 and PS2 dependent photosynthetic efficiency. So what is the yield of these, PS1 and the PTR? This is only shown, PS2 is only shown the electron transport rates. So you can do all that, and ultimately you can consolidate the data in the form of PCA, where you can find out which factor is contributing more, how they are interacting, etc., etc. So we studied, all these growth parameters which you see here, or CAT or GPX or yield or photosynthetic efficiency or seed proteins, calories, lipid oxidation, SOD, hydrogen peroxide, soluble sugars, and so on, multiple parameters. And many of these are regulating different pathways in the plants. For instance, again, an important point, when the plants are challenged by pathogen, usually the phenylpropanide pathway leading to the production of whole lot of metabolites which are together called as secondary metabolites is activated. That is, some of them are called as phytoreactions also, which are responsible for countering the pathogen when infects, and is able to eliminate pathogen. So we studied the enzyme of serine protonite pathway as well, for instance, leading to the production of the monolignols. Lignin is again another important component. If the plant is more lignified, less possibility of pathogen, is more stress resilient, chalcone synthase, anthocyanins, and a lot of the products like flavonoids, polyphenols, etc. And then we also studied the expression of certain proteins like PRB, which I mentioned, of them the prominent being beta-1 glucanase, responsible for loosening of the plant cell wall, and chitinase responsible for dissolving the fungal cell wall so that the attack is weakened. So both these enzymes. So gene expression of these enzymes, and then again PCA for all this, understand is catechols, phenylpropanides, lipoxygenase, phenylalanine ammonia-lyase, GST, salicylic acid chitinase and glucanase. And ultimately, I'm coming to the conclusion because I have to take another story also, just to that is very important. I'll expose to the other set of stories as well. So stress. So this may improve ultimately, or what were our conclusions? Improve the nutritional quality of seeds by accumulating stress-induced proteins and antioxidants. So your seed gets enriched in quality in terms of the proteins. A very, very important nutritional trait of the seeds. The second point, predisposing plants to drought stress, followed by application of an elicitor. Elicitor is a molecule which behaves similarly when fungi attacks the plants. So it can mimic the action of fungi. So drought followed by biotin in terms of artificial elicitor that causes induction of biotic stress. And this again is very interesting. So this as well can lead to, depending on the sequence, the order in which it is done, to produce protein and antioxidant rich seeds. So of course they vary. These stress responses can vary depending upon what are the environmental conditions and the cultivar, the original cultivar which you've taken. And then I talked already of synergism and antagonism between the SA and jasmonic acid regulated signaling pathways. And PR proteins, interestingly, is again very, very important discovery by us, that PR proteins, which were hitherto supposed to have been produced only when plants were challenged with pathogens, were also found under their abiotic stress like drought. So this again is very important. That means PR proteins are also produced under drought. Phenylpropionate pathway I mentioned, it was upregulated because if it is challenged, it needs to produce more of the secondary metabolites. So it was upregulated, whereas the glucanase and chitinase expression was suppressed when you provide the combined stress. So a cultivar could be more tolerant towards individual stress, but might perish under multiple stress conditions. Fair enough, it is understandable. But then already stress plants switch their defense strategy on encountering another stress condition. For example, I have a crop which is challenged by drought. Now, if I challenge it by fungal pattern, there is an immediate switch in its defense strategy. Entire machinery, biochemical pathway, gene regulation, etc. is changed. So that is what happens. So this is one part of the story which I thought I'll share with you, and I hope it helps you. You are able to understand. It's a major area of research. Thousands of scientists are working on this globally. So it's a very important area because it can address the problems of the food security. It's a major problem. Okay. Another point, which another thing, of course, all this is all well received. Another point, which I thought I'll share another 10 minutes or so I'll be able to finish, because I'll just touch on the important aspects of that. Another challenge, another problem, major problem, which we experience in our food technology colleagues are also sitting there, and they will also appreciate this, that post-harvest loss of the crops. It's to the tune of nearly 25%. That is the crop, you have the harvest, you have harvested the crop until it reaches the consumer. There is a loss to the extent of 45%. And it's really a great problem. It a lot of challenges. There could be many reasons. You are aware that government talks about storage conditions, etc., etc. Transportation is a loss. So all those factors are responsible. Handling, lack of market access. If it is lying in the mandi for 3 days, then it gets destroyed. There are no buyers. A lot of things, lot of contributions, and then we have to spend money to increase the shelf life, refrigeration, storage, heat treatment, modify the atmospheric packaging, 1-methyl cyclopropane, calcium chloride, sulfur dioxide. I mean, these are some of the strategies with which increase the shelf life of the harvested crops. Electromagnetic radiation is important. You are aware. So, discoverers range two types. You are aware. I don't think I need to give an introduction. Non-ionizing and ionizing. Microwave, I have put red here. Non-ionizing radiation. The frequencies, it ranges from 300 megahertz to 300 gigahertz. And there are different characteristics of the microwave. Microwave, you are also using so the microwaves now, and the microwave set again, because the next figure which you see is thermal effect, non-thermal. So the thermal effect we are using for houses. In houses, we are using the thermal effect. So two different types. If the specific absorption ratio, which is called as SAR, is less than 0.1 or more than 1, then it will produce different effects, thermal and non-thermal. Non-thermal effect has been exploited, as well as been exploited, because it's responsible for the nutritional value is largely maintained. There is no formation of undesired compound, gentle processing, etc., etc. Now, in plants as well, this has been exploited. But in what? It has been exploited, for instance, in seed germination processes, in photosynthetic efficiency, to enhance the photosynthetic efficiency, to enhance the antioxidant efficiency, for heat management, and for brain disinfestation. So it already has applications, but we exploited this to enhance the shelf life of the crops, of the harvest. For instance, and we worked on, tomato. Tomato, in fact, incidentally, there is 12%. If I talk of the Indian figures, there is a 12% post-harvest loss of the crop, of the produce of the tomato fruit in India. So a huge loss. It is number 2. The first one is guava. In guava, you have the higher post-harvest loss, and the second is potato. Second is tomato. So we try to address this problem, that what can be done about reducing this post-harvest loss of tomato. So our objective was to enhance the food shelf life with increased nutritive value. So twofold objective. And for that, we did a lot of investigation of a lot of other parameters as well. And we undertook this study in 2 different phases. One is that you have the vegetative phase, where you have the tomatoes. You expose them to a specific dose of microwave radiations. And second, once the tomato fruit is ready, second stage, you give a second dose of microwave radiation. So starting vegetative in seeds and in fruits. And we took 2 varieties, one in Kharif and Rabi season. As you know, there are 2 important seasons. And this is the experimental layout, how it was done, initially exposed. And the seeds were exposed. The left side one, on the left side, you see the seeds are exposed, and then the plants are grown out of the exposed seeds and the tomato which are grown are harvested and then they are irradiated again, second stage. Of course, you have to ensure a lot of things that there is a specific homogeneous distribution of hybrid variation all over the sample called as SAR, etc., etc. So we studied all those parameters. And we did a lot of work on also dose optimization, which, how many hours it should be exposed with how many frequency, etc. etc. And then we studied the germination percentage, germination rate, germination index, beta 1 logarithms, activity, and a lot of other growth parameters and the biochemical parameters, bioactive compounds like polyphenols, flavonoids, or TPC. And also we undertook to study the structural changes, the Raman spectrum of the FTIR, and did a lot of such investigations also for phenolics, beta carotenes, lipids, carbohydrates, just to see what is happening in their biochemistry, phenolics, lipopene. So, and also their photosynthetic efficiency of the plants when they were flowering, at the second stage. I mentioned about dual bands, so we use the same technology. And then the phase 2, called as the breaker stage for what the breaker stage is, that when the tomato skin is still green and it starts turning red. So that is called as the breaker stage. So the second dose which we gave was at the breaker stage, green but starting to turn red. And pericarp is very crucial. What is pericarp? Pericarp is the fleshy part of tomato, inside tomato. So that is again, if you are able to prolong its degradation, then naturally you can increase the shelf life. So it is very important. Pericarp. And we did, to investigate the pericarp changes, we did, FESM analysis and expose them to different doses of electromagnetic radiations through the, different doses like SAR1, SAR2, SAR3. SAR1 and 2, they are non-thermal, and SAR3 was thermal. So we introduced a variant now at this stage. And so 3 factors. Ultimately, at the end of everything, 3 factors contribute to the shelf life of tomato. What are those? Weight loss, rapid weight loss, or extended weight loss. It will contribute to the maturation. Pericarp, I already mentioned, pericarp firmness. Again, very important. The fleshy razor. And ethylene. Ethylene is a hormone which enhances the maturation. A very important hormone. So we investigated further these 3 parameters: weight loss, cell wall degrading enzymes. For that, we undertook 3 enzymes: polygalacturonase, responsible for the degradation of polygalacturones, which are forming pericarp. Then beta galactosidase, responsible for degradation of the galactons, which are again polysaccharides present in the pericarp region of the tomato, and pectin methylestrate, responsible for degradation of pectins, which is again a component of the tomato fruits. So all those parameters, and we found that exposure worked favorably for all the parameters which we tested. I will not go into the detail. We also undertook the gene expression of 2 enzymes. Ethyl I mentioned. So there are 2 enzymes, ACC synthase and ACC oxidase. These are the enzymes of ethylene biosynthesis pathway. So it was important to see that how these enzymes are reacting when they are exposed to electromagnetic radiations. So carboxylic acid synthase and aminocyclic carboxylic acid oxidase. These 2 important enzymes. So we undertook their gene expression analysis, and finally come to the conclusions. This probably is one of the last slides. So we, of course, microwave radiation parameters, ensure uniformity, exposure time, tested a lot of parameters. Seeds, germination percentage, the germination, index. So they increased. That is promotes the effect of the microwave radiation. Beta-1,3-glucanase is the enzyme responsible for degrading glucans in the seeds. That means if glucans are degraded fast, seed will germinate fast. So it also goes up. Biometric, bioactive compounds, polyphenols, total protein, they all increased. And 4 hours we found the stimulatory effects for seeds as well as for the flowering stage. So finally, on the right side, you see is microwave non-radiated tomato fruit, and on the left you see microwave radiated. So in microwave radiated, photosynthetic efficiency goes up in the growing plants. There is a compositional change of the proteins lycopene, which is one of the carotenoids, and bioactive compounds. There is a less weight loss, reduction weight loss, contributing to increased shelf life, cell wall degrading enzymes, all three I mentioned, and ethylene genes. They are down regulated. That means ethylene biosynthesis is delayed, therefore delaying the maturation of tomato, I think. So, post-harvest application, which I mentioned, of non-thermal, because we also took thermal. Non-thermal microwave is more useful. It increases the shelf life, and once the technique is available, it can be naturally utilized, because a lot of is a general problem, not just with tomato, guava also I mentioned, and a lot of other fruits, and it's a major problem leading to up to 40% of the problems. So this again is of course published as Computer and Electronics in Agriculture and post-harvest biotechnology in 2 different journals, but I have not put them here. But I think then with this I will conclude. So my entire idea was to give you an exposure, not going into the deep nitty-gritties of the research data, but to expose you to 2 major problems, which our present crop productivity is severely affected. So I hope that the message was gone down well, and you can think about it, and you can contribute maybe in future and may come out with a certain practice in this area. If you have any questions on this,
Yes. Any question, anyone? Next students, what more of your area is the post-harvest technology?
you can ask now, because advantage would be that if you ask, then the other audience is also benefited. If you come to me later also, I have no problem, but then a lot of other audiences also there. If you ask such questions, then I think a lot of other of you are listening, and they will also benefit. But it doesn't matter. Okay.
One question from my side, the fungal pathogen, how we can actually mitigate those problems? The notorious fungal pathogen, which you had mentioned.
A lot of strategies are biocontrol.
Yeah, that's what I was actually thinking about.
Biocontrol is very important.
Yeah. No question.
Okay. I think it's okay. आपका दूसरा lecture भी तो होगा ना, बहुत short में उसको.
जी जी.
ऐसे
25 minutes ago. 30 minutes. Yes. Thank you, sir. Thank you. Thank you so much, sir, for the insightful lecture. Our next speaker already have joined us. So without much delay, I would like to introduce our next speaker of the day, Professor Dr. U.K. Madhusudanan. Professor Dr. Madhusudanan is a distinguished biomedical scientist and additional professor in the Department of Biochemistry at Sri Chitra Tirunal Institute of Medical Sciences and Technology, Kerala. His research lies at the critical intersection of neurogenetics, molecular biology, and translational biochemistry, with a specific focus on rare neurodegenerative diseases. Dr. Madhusudanan's work aims to uncover the genetic and molecular underpinnings of neurodegenerative disorders, including those with rare presentations, thereby contributing to the development of blood-based biomarkers and novel biochemical assays for early detection and monitoring. His research bridges laboratory science with clinical relevance, offering potential for impactful diagnostic innovations in neurology. He has authored and co-authored numerous peer-reviewed publications in reputed international journals. Those of you who are having classes right now, final year students as well as the 5th semester students, they are supposed to be here. All the final year as well as the 3rd year students, they are supposed to be here. Otherwise, your attendance will not be sent to the respective faculty. Come on, settle down fast, everyone. Let me continue the introduction. Professor Madhusudanan has authored and co-authored numerous peer-reviewed publications in reputed international journals. His work has been explored through academic databases under the author name Madhusudanan UK or Madhusudanan Urulangadi, and reflects his scholarly contributions to the fields of clinical biochemistry, neurobiology, and molecular diagnostics. At Sri Chitra Tirunal Institute of Medical Sciences and Technology, Kerala, Dr. Madhusudanan is also actively involved in mentoring young scientists, leading collaborative research initiatives, and contributing to the advancement of diagnostic technologies relevant to public health. Professor Madhusudanan, over to you. Will you be able to share your slides?
Yes, thank you. Can you hear me now?
Yes, yes.
Okay, let me share my slides. Is it visible?
Yes, it is absolutely fine. Can you please put it in the slideshow mode?
Yeah, it is in slideshow.
Yes, it is. It's visible full screen now. Yes, thank you.
Yeah, it's in full screen. All right. Okay, very good morning to everyone, and I will move from the plants to the human diseases right now. So as Dr. Vijay has already mentioned, I work at the Department of Biochemistry at the Sri Chitra Tirunal Institute for Medical Science and Technology, which is a tertiary care center for the diagnosis and treatment of cardiovascular as well as neurodegenerative diseases. And it is under Department of Science and Technology. And most of my research work is trying to understand the fundamental principles of rare neurodegenerative disease as well as the disease mostly affecting the brain. So today, although my title has many factors, but I will just focus on 2 aspects of the neurodegenerative diseases. So as you all know, the neurodegenerative diseases are progressive and mostly associated with aging. Many of the neurodegenerative diseases. And there's a progressive loss of structure or the functions of the neuron. For example, the Alzheimer's disease, Parkinson's, Huntington disease, or amyotrophic lateral sclerosis. And most of the disease share some of the common features as well as they have independent presentations. For example, the protein aggregation and formation of the aggregated proteins accumulation in the neurons has been observed in most of the neurodegenerative diseases, and oxidative stress and mitochondrial dysfunction along with the lysosomal dysfunction has been uh, implicated in the both the disease occurrence as well as the progressions. So some of the common symptoms include the memory loss and motor dysfunction, and cognitive decline is associated with most of the neurodegenerative disease. Very interestingly, or both the treatment as well as the diagnosis of many of the diseases are early stages. It is very difficult because the neurodegeneration happens over a period of time. So the early diagnosis is very difficult many times due to both the unavailability of the effective diagnostic test. Unlike diabetes or hypertension, we have a very simple test to do it. But for neurodegenerative diseases, it is very difficult. And rare, neurodegenerative diseases are actually presents with a similar sort of symptoms like the developmental delay, language problems, and the low IQ. But it is very difficult to diagnose due to the reasons for the development of the diseases. Although the genetic screening is there, but genetic screening is also expensive, and sometimes it is not feasible until the symptoms appears. So this is a big challenge. And in my laboratory, although I work with a lot of neurodegenerative diseases, rare diseases, but one of the main focus is Parkinson disease. Although it is not very rare, but it is still considered as rare under WHO classification. So I will present, so in my laboratory, the major focus is written here, like the exosomal micro, I will come to that a little bit later, exosomal proteins, and the genetic architecture of Parkinson's. So we have been working with a big collaborative pan-India genomic projects on genetic basis of Parkinson disease. And also I work with a lot of functional assays like the lysosomal functional assay where glucose cerebrosidase activity can be measured accurately, and the neuronal DNA repair and cell death mechanisms, mitochondrial energetics, and the plasma lipidomics and metabolomics for understanding or to getting some differentially regulated factors, which ultimately can be useful both as biomarker as well as for the disease pathogenesis. So this is the main theme. One of the main theme I work is on Parkinson's disease. In addition to that, there are metabolic disorders and mitochondrial disorders. We work with a lot of mitochondrial disorders, rare disorders. Mostly they are genetic in the mutations in either in the nuclear gene or in the mitochondrial gene. For example, the coenzyme Q biosynthetic pathway or electron transport chain problems, and metabolic disorders like the lysosomal storage disorders and so on. So what is Parkinson's disease? So this is the second most common neurodegenerative disorder after Alzheimer's disease. And the main symptoms are affecting the movement of the body. So it is called a movement disorder. It affects the motor symptoms. For example, the patients who has Parkinson's disease has bradykinesia, means less mobility of the legs and hands, rigidity, postural instability, resting tremor. And these are very common symptoms, but the patient, when they come to the clinic, they have all these symptoms, which means that at least 10 or 15 years back, the neurodegeneration has started, or even before. And they are also present with a lot of non-motor symptoms like sleep issues, olfactory dysfunction. The loss of smell happens much years before the symptom actually appears, and gastrointestinal changes, and they undergo a lot of psychological issues, depression, constipation, and so on and so forth. And the major clinical presentation, pathological presentation, is the accumulation of alpha-synuclein aggregates in the form of Lewy bodies, which accumulates in the substantia nigra region in the midbrain. So there is a selective loss of neurons from the substantia nigra region. Okay, so there's a degeneration. So that is the main reason. And the causes are many. So there are mostly associated with aging. As the age progresses, so along with all the body function starts declining, and there is a neuronal decline as well. And there are at least 10 to 15% of the disease is caused by genetic mutations, like the alpha-synuclein gene Parkin, LRRK2, GBA. These are the mutations occurs in many patients. And also so only 10 to 15% are genetic. The remainings are sporadic or they are caused by unknown factors. So it has been reported that there are causes like environmental risk factors, mostly pesticide exposures like rotenone. They are used as herbicides and pest control agents. And the rural living, even there are some epidemiological studies which shows that even the drinking the well water, heavy metals, these are all can lead to Parkinson disease. But ultimately, all these factors, either genetic mutations, aging, or environmental factors, they all affects either mitochondrial function, the increased oxidative stress, defective autophagy, inflammation, and ultimately protein aggregation, and then the neuronal cell death, which leads to the appearance of the symptoms. So currently, so this is the disease progression. If you look at the pre-symptomatic phase, that we do not know exactly when it starts, and that time there are no symptoms, but the neurodegeneration slowly starts. So the moment the patient reach clinics, almost 80% of their neurons have already lost. Okay, and there are non-motor symptoms appears like the sleep disturbances, and there are a little bit of non-motor symptoms like olfactory problems, mood swings, autoimmune systems. And the specific symptom starts much later. By the time the motor symptoms starts appearing, motor symptoms like movement issues, only the diagnosis happens at the last stage. So that means maybe 10 or 15 or 20 years before, already the neurodegeneration have started. Okay, so there is no way one can diagnose this, and other than genetic screening is possible, but genetic causes is only 10 to 15%. So most likely we are going to miss out all the diagnosis. Okay, so this is a major challenge. And currently, unlike many diseases, Parkinson disease is diagnosed clinically. There are no biochemical or imaging markers that can actually predicts or diagnose. So it is still a clinically diagnosed condition. There are different criteria like the Movement Disorder Society, UPDRS, and the Hoehn and Yahr scale. So these are all different stages. Okay, so different stages of the disease can be diagnosed, but there are a lot of limitations with the clinical diagnosis because they are very subjective and all the patients may not present all the symptoms. Okay, there is a stage dependent which stage they are reached, and it's not very predictive, and there is no insights into pathology. So we don't get any of this information with the clinical diagnosis. So there are other diagnostic methods like the imaging, genetic, and the body fluid biomarkers, like imaging like DAT SPECT, PET, or MRI scanning. Although we can directly visualize the substantia nigra degeneration of Parkinson disease, but it cannot predict. And doing an MRI, PET, or SPECT in a healthy person is very unlikely going to happen. And it also helps to distinguish from non-degenerative trauma. There are also associates. There are also Parkinson disease, and there is a group of disorders called Parkinsonism disorders. Their symptoms appears like Parkinson disease, but they are not. So this differential diagnosis is possible with this imaging because we can actually look at where the degeneration happens. But anyway, this happens only after the disease occurs. There is no predictive marker. And there are genetic factors, as I mentioned, there are only 10 to 15% of the Parkinson disease are genetic. So this is possible to do genetic screening because it is expensive, and without indication, people usually in India will not do a genetic testing. And there's like LRRK, GBA, PARK7, PINK1, alpha-synuclein, all these mutations can be screened. There are also a lot of research ongoing to look at the body fluids as marker because these are minimally invasive procedures. You can get the blood, urine, saliva, to some extent CSF. CSF is not possible because it's an invasive procedure, but there are factors like the total alpha-synuclein or its phosphorylated form can be used as a diagnostic marker, and the DJ1 and the neurofilamentin protein, light chain protein. So these are all have been used before, but there are a lot of contradictory results because these are not absolutely used as a diagnostic test. Still, the clinical diagnosis is still ongoing. Okay, and there are also plasma, urine, or serum biomarkers like DJ1, plasma uric acid, tau proteins, amyloid beta. These are all has been used in Parkinson disease for the diagnosis, but these are not a universal markers to use, can be used. There can be an accessory markers, but they are not definitive for early diagnosis. So this slide show you the general limitations of the currently available Parkinson disease biomarkers. So they have a limited sensitivity in the early stages. It cannot differentiate atypical Parkinsonism because, as I mentioned already, there are similar symptoms, but they are not Parkinson disease. So you cannot differentiate all these subtypes or similar symptomatic disorders based on the currently available markers that I have discussed so far. And many are invasive and expensive. For example, the imaging is expensive and the genetic testing is very expensive. And also genetic testing is not used for sporadic PD because only 10 to 15% are genetic. Remaining 85% are sporadic. That's not due to genetic cause. So that also makes genetic testing, unless one has to do a family screening, it is possible. And the limited correlation with the disease progression. So none of the markers, as I mentioned before, cannot predict the disease progression. For example, alpha-synuclein has been used extensively, but there is a problem because alpha-synuclein is present in the RBCs and platelets. So that will cross-contaminate, and whatever results you get is due to contamination. It may not be actually reflecting the brain pathology. And there's a lack of brain specificity because many of these factors which I mentioned, like the DJ1, all these proteins are not very brain specific. So there is no brain specific markers that can be used. So we move on. So all these things, because most of the biomarkers which I have mentioned has been directly measuring in the plasma. So there's an excellent way we can use is the extracellular vesicles or exosomes. So this is an alternative source of biomarkers. There's a lot of research work is ongoing in India and abroad on extracellular vesicles. So these are EVs. It's called EVs. They are very nano-sized vesicles of 30 to 150 nanometer. And they are originating from the intracellular. Okay, they are originated from the endosomal pathway, and they are secreted by all types of cells, both in physiological and pathological states. So the content of the exosomes actually reflects the which host cell or tissue or organ which comes from. So in general, they carry functional molecule. That's important because they can carry DNA, messenger RNA, microRNA, proteins, lipids, metabolites, and they have been used for a cell-to-cell communication. And the interesting thing is that they can cross the blood-brain barrier. So the brain-derived extracellular vesicles can be measured in body fluids like in blood. And they are detected in all body fluids, and the cargos are stable because there's a lipid bilayer. So it's like micelles or the lipid bodies that it has inside all the contents, most of the proteins and microRNAs and DNA. So this is an excellent model that can be used as a biomarker discovery, and it can cross the blood-brain barrier. So we expect that most of the disease, neurodegenerative disease, for example, Parkinson disease, whatever brain pathology happens, those proteins or microRNA or factors or metabolites can be detected inside. So in that way, we can understand both the biomarker level as well as the disease pathology. Okay, so today, this is the workflow which I'm going to talk today. So we collect blood from the Parkinson's and the controls, like healthy controls, and we do directly the untargeted metabolites, or we isolate the plasma extracellular vesicles, and we also have a way to isolate brain-derived extracellular vesicles, and we have been doing a microRNA profiling to understand the differentially expressed or differentially regulated microRNAs within the exosomes, as well as untargeted proteomics to get an idea of what are the proteome differences between the Parkinson disease patients as well as controls. Okay. So coming to the proteomic study, so what we do, the isolation of extracellular vesicles is not so easy because they are very small, like 50 to 150 nanometer. So what we follow is the differential ultracentrifugation. So stepwise increasing the speed and multiple levels, we remove all the cellular debris because most of the time the contaminant comes is the HDL cholesterol, LDL cholesterol, all this blood and the albumin. So we systematically, step by step, we perform the ultracentrifugation. Like the last speed is at the 1 lakh G for 70 minutes. And then we finally dissolve the whatever extracellular vesicle we get, and then we give for a mass spectrometry to analyze the proteome. Okay, so before giving the proteomic analysis, we need to confirm that whatever we get is extracellular vesicle. So initial characterization of extracellular vesicle we do with a transmission electron microscopy. So you can see that there's a shape, this is a typical shape of an extracellular vesicles of around 100 nanometers, both in the control population as well as Parkinson disease. So these are isolated extracellular vesicles from the plasma from both the controls as well as the disease patients. And we also do a dynamic light scattering. And you can see there is a single homogeneous peak which corresponds to around 80 to 90 nanometers from both the healthy controls as well as the disease. So whatever we isolated extracellular vesicle showed a very characteristic shape by transmission electron microscopy, and the size distribution is very heterogeneous. So which in this way we confirm that whatever we have is a homogeneous preparation of extracellular vesicles. We further confirm it with the Western blot analysis because there are very specific extracellular vesicle markers like TSG101, HSP90, because these are present in almost all the extracellular vesicles regardless of their origin. So they are like TSG. So you can see both in the controls and the disease, the presence of the marker TSG, and albumin is a copy. Albumin comes along with the extracellular vesicles from the plasma. You can see here, and HSP90, and Calnex is used as a negative control, which is absent in extracellular vesicle, but in cell lysate they are present. So this is used as a negative control, further confirming that there is no cellular contamination, but you have a pure extracellular vesicles. So they showed a character. So then the question comes whether, because these extracellular vesicles are isolated from the plasma. So before going for proteomics analysis, we wanted to confirm that our total extracellular vesicle has brain-derived EVs as well. So there is a very specific marker. It's called L1CAM or CD171, which is a brain-derived EV marker, because most of the brain-derived extracellular vesicles will have L1CAM in the extracellular vesicle membrane. So in this Western blot, you can see that both in controls and disease, there is an L1CAM is present. So which suggests that in our preparations, there are some percentage of EVs are actually derived from the brain, so that our proteomics will give an idea what is the changes happens in the brain. Okay, then we give an proteomic profiling. So what we did was we isolated from both the PD patients as well as controls. We performed the extracellular vesicle isolation, as I mentioned before, by ultracentrifugation, and when we gave for mass spectrometry analysis, and we have actually, after a lot of filtering and analysis, we have identified 65 very unique proteins which are differentially present in the Parkinson disease. And after further, we did a lot of bioinformatics as well as cutoff analysis, and we found 58 proteins were actually the response. The remaining few proteins are very ambiguous, so we removed all those proteins, and finally we analyzed 58 proteins from the proteomic analysis. So among that, there are around 14 proteins are very uniquely regulated, like the caspase, clusterin, zinc finger protein, serotransferrin, and coffulin. So these are very interesting proteins because there are some literature which suggests that these proteins have some pathological role to play in the development of Parkinson's disease. So one by one, we started analyzing both looking at the biomarker potential of these proteins as well as their relation to the disease pathology. So if you look at, what we did was, there is already literature available, the databases are available, which are called Vesiclepedia as well as ExoCarta. So they deposit all the studies on extracellular vesicles, and a lot of proteins are listed. And we took the top 100 proteins and we compared with our proteomics data. Okay, so when we compare, these are the Vesiclepedia proteins, and this is the ExoCarta proteins, and this is our protein, 58 proteins here. And what we found was there's only one common protein is found, that is the coffulin-1. So coffulin-1 is a protein which is involved in the actin cytoskeleton regulation. They are involved in cell motility, cell mobility as well. And there are few literature which suggests that coffulin-1 has very important role in neurodegenerative diseases, because they are very important for neurotransmission, migration of, or the movement of mitochondria and vesicles from the axons to the dendrites, and there are a lot of functions. And very interestingly, so since only one protein was found very common between the databases, we wanted to further analyze about it. So the heat map also shows that the coffulin 1 is differentially regulated, and this is increased in Parkinson disease. And most of the proteomics most of the proteins which we obtain through proteomics are involved in extracellular vesicle, cytoskeleton. Majority of the proteins were cytoskeleton, like 31% of the proteins are cytoskeletons, extracellular regions, and few other functions. So, and the string analysis also showed that there are the biological functions of these proteins include complement binding, opsonin binding, chaperones, and the protein binding. So all, and also the constituent of postsynaptic actin cytoskeleton. So coffulin 1 is involved in this pathway. So in string analysis as well, we found that this protein is really involved in actin cytoskeleton regulation, and they can be potentially involved in disease pathogenesis. Okay. So we further validate the coffulin 1 in a different set of disease groups. So what we freshly isolated extracellular vesicles from both the control 80 and the Parkinson disease 91 patients. And we also used two other diseases, PSP. PSP is actually a non-Parkinsonian disease, but the symptoms are very similar to Parkinson disease, and also amyotrophic lateral sclerosis. This is actually, if you want to detect a biomarker, if coffulin 1 is very specific to Parkinson disease, we need to compare other disorders. So we use these group of disease patients samples as a control to check whether coffulin 1 is very selective for Parkinson disease. Okay. So you can see in this picture, when you compare extracellular vesicle coffulin 1 by ELISA, we used ELISA here. So we have taken patients and controls, and you can see that in Parkinson disease patient, there is a very selective increase in very statistically significant elevated coffulin 1. So which suggests that this can be used potentially as a disease marker. And you can see in the ROC curve as well as the precision recall curve, their sensitivity and specificity are 93 and 85%, which means that they are very good candidates to be used as a biomarker. Okay. So we further validated with a different set of patients and different groups, early stage, middle stage, late stage, etc. And this coffulin 1 seems to be very interesting protein, and it shows very selective increase in Parkinson disease. And we also compared, sorry, I will come to the next slide. So this is the comparison with related movement disorder like the PSP, which is a non-Parkinsonian disease, but the similar symptom. But you can see that when you see the control and PSP, there is no significant decrease. So which suggests that this coffulin 1 could be a very selective marker for Parkinson disease, and they can be used to discriminate Parkinson disease from other related disorders. Okay, so the next question. So whatever we isolated is from the plasma. So we need to check whether these extracellular vesicles are actually derived from brain. So what we used was, we used an L1 CAM dependent immunoprecipitation. Okay. So you can see that these are the input total extracellular vesicles, plasma EV. And when we do an immunoprecipitation using L1 CAM antibody, this is a magnetic bead based pull down. So where you can very selectively pull down brain derived EVs. So you can see that in the brain derived EVs, the coffulin 1 is highly enriched. So which suggests that coffulin 1 is in fact derived from the brain. Okay. So so you don't need an invasive procedure like the collecting the CSF, but you can get the brain specific proteins in the plasma itself. That's the conclusion of this slide. So we further want to confirm that whether coffulin 1 is actually present in the brain. Okay. So what we did, so we cannot get a human brain naturally. So we made a animal model. So there's a mice brain model. And then we did an immunofluorescence, and you can very clearly see that the coffulin 1 is in fact present in the substantia nigra region of the brain of PD mice. So which suggests that extracellular vesicles which carries coffulin 1 from the neurodegenerated brains of the PD, and it crossed the blood-brain barrier, and these extracellular vesicles we can detect in the plasma, and this can be used as a potential biomarker. So to conclude this part, so we can see that the one of the protein we have identified through our untargeted proteomics, and we could get at the moment one protein which very likely to be a very specific marker for Parkinson's disease. So now currently we are further studying in more number of samples, and and we also want to see whether, as I mentioned before, there are different stages of the disease: early stage, late stage, and the pre-stage. So we are trying to get group of people in all these groups, and we want to check whether this can be used as a early diagnosis marker. So that work is ongoing. So now I come to another small part. So we also, along with the protein, we also did metabolomics in the plasma. So we have selected another set of group of patients, and we did untargeted metabolomics. And what we have identified, there are a lot of changes in the metabolomics between Parkinson's disease and controls. So you can see tyrosine metabolism, phenylalanine metabolism, and the lysine, most of the amino acid metabolism, tryptophan, arginine, biotin metabolism, all these things has been extremely increased in Parkinson's disease plasma. But very interestingly, we are now following up sphingolipid metabolism, because sphingolipids are one of the major components in the myelin and the neuronal cells. So we analyzed, and you can see that there are a lot of so this orange color is the control, and the bluish, this is the Parkinson's disease. So you can see that many of the proteins like sphingomyelin, sphinganin, sphingosin, all these proteins are actually differentially expressed between controls and the patient. So this is one of the interesting factors which now we are trying to explore. And there are many very specific proteins like the ceramides, sphinganin, sphingosin, and all these factors has been significantly different between the Parkinson's disease and control. So untargeted metabolomics also can be used for many of the neurodegenerative disease to understand what is happening in the brain neurology. So ultimately, this can be either used as a biomarker, or this can be used as a way to understand the disease better. In that way, one can identify new drug targets, for example. Okay, so if you look at the sphingosin metabolism, so there are two pathways. One is the de novo pathway, and one is the salvage pathway. So we are now trying to understand all this, because these pathways has been extensively implicated in neurodegeneration. So we were trying to understand whether the de novo pathway is affected, or the salvage pathway. So salvage pathway, they reuse the brain material, like reuse the membranes and lipids used for the building of the new materials. But in de novo, they use amino acids and the palmitoyl-CoA to synthesize sphingolipids. So now we are step by step, like individually, we are trying to understand, based on our metabolomic results, we have some hints that there are which pathways altered, which pathways differentially expressed. So we are looking at the mRNA level, we are looking at the individual genes as well, and we are also looking at the metabolites, intermediates. Okay. So to summarize, what we found was the glucosyl ceramides, which is a sphingonin, this is a sphingomyelin, suggests that there is a fundamental disruption in sphingolipid metabolism in Parkinson's disease, which most likely due to lysosomal dysfunction. That I did not go in detail, but anyone interested, you can study the salvage and de novo pathway of sphingolipids, and they also increase the ceramide metabolism. Okay, ceramide 1-phosphate, which suggests that there is this ceramide 1-phosphate in literature shows that they can induce apoptosis and inflammation. So as the age increases, then there's a decline in the lysosomal function, for example. So this can lead to altered lipid metabolism, and that can actually induce inflammation and apoptosis, and ultimately leads to neurodegeneration. So this is the line of thinking that we are now looking at, and that the work is ongoing. Okay, so ultimately, so I have presented two pathways. One is the proteomics and extracellular vesicle pathways involved in Parkinson's disease as a biomarker, as well as the metabolomics, understanding the lipid metabolism in Parkinson's disease. And genomics, we are actually currently doing a lot of genomics work to understand throughout India what are the genetic architecture of the disease. And along with that, I am also working with biochemical assays to diagnose or to understand the severity of the Parkinson disease. And mitochondrial work we have been doing a separate project. So ultimately, all this we are looking at the genomics, we are looking at the metabolomics, we are looking at the proteomics, and we are also looking at functional assays. We are looking at the mitochondrial assays of Parkinson disease. Okay. So, so I will stop here. I think it's 45 minutes. So most of this work is the EV extracellular vesicle proteomics is done by the PhD student Deepthi, and metabolomics is worked by another PhD student Priya. And this work is actually done with collaboration with our own institute. Like Professor Shyam is a professor at neurology, and he diagnosed the disease and he gives us the sample. And Dr. Poonam Thakur from ICGETEI Trivandrum, and she is collaborator for multiple projects. And for this particular project, she has the animal model, mice model of Parkinson disease. So most of the staining and the neuroimaging, everything is done by her. And the Rajiv Gandhi Center for Biotechnology, he does the proteomics and the metabolomics for me. That's a facility where we submit the sample and we get the result. And also our own department, my HOD. And the particular work which I mentioned today is funded by ICMR. So there's a work is going on. Okay. So thank you so much. If you have any questions, I can answer now.
Thank you, Professor Madhu, for such a wonderful and informative presentation. We have gone through the every stages of Alzheimer's disease. That's really wonderful to know, and how we should actually be proactive in diagnosing it early so that we can have good outcomes for the patients rather than suffering for the entire life. That was really insightful. Now I'll open the questions to the audience. Anyone would like to ask any question? Nobody. By the way, no questions from the audience. They seems to be sleeping in the morning itself. Is it? If there are any questions, I will get back to you, Professor Madhu, through your email, if that is fine with you.
Yeah, perfect. That's absolutely fine.
Okay. And thank you so much for such a
Thank you
wonderful
so much.
presentation, and thank you for joining us today morning.
Thank you.
Cool.
Yeah.
I know you are very busy. You have to catch a train soon, so
I'm that's absolutely fine. So anyone wants, they can get my email ID from the website. If anything is important, you can discuss,
Yes.
Yeah. Thank you.
Sure. Thank you.
Thank you.
Our next speaker of the day, Professor Ragini Singh. She is already online, so I'd like to take the privilege of welcoming Professor Ragini Singh. Professor Ragini Singh is a dedicated biomedical researcher and academician, currently serving as additional professor in the Department of Biochemistry at All India Institute of Medical Sciences, Rajkot. With a strong foundation in molecular biology, cancer research, and clinical biochemistry, she brings over a decade of experience in translational research and medical education. Dr. Singh has received advanced training in cytogenetic and molecular diagnostics of genetic disorders at All India Institute of Medical Sciences, New Delhi, and received advanced training in cytogenetic and molecular diagnostics, and also specialized training in genomics and proteomics from the Tumor Biology Centrum, University Klinikum Eppendorf, Hamburg, and Germany. She was associated for over 12 years with the Gujarat Cancer and Research Institute, Ahmedabad, contributing significantly to cancer biology research. Her professional development includes prestigious certificate programs such as the Basic Course in Biomedical Research, ICMR, and a current training in data science and decision science at IIT Delhi. Dr. Singh's academic excellence has been recognized with several honors, including KPS PS Sinha Award for the best paper in the Indian Journal of Clinical Biochemistry, Landmark Paper Award for her publication in oral diseases, National Scholarship and Bhamashah Award as a university topper at the undergrad level, internship scholarship by the University of Sharjah for training in systemic reviews and meta-analysis. She currently leads multiple intramural research projects focused on salivary biomarkers, IL-6 and IL-8 for oral cancer detection, lipoprotein A profiling in children of the Saurashtra region, fetal hemodynamic monitoring during labor, integrating AI in undergraduate bioinformatics education. Dr. Singh also holds key academic responsibilities, serving as a member of the Institutional Ethics Committee at All India Institute of Medical Sciences, Rajkot, and guest editor for special issues of Critical Reviews in Oncogenesis. Her core research interest areas include oral cancer biomarkers and molecular pathogenesis, innovations in diagnostics and therapeutics, public health and policy impact, and with her blend of clinical insight, laboratory excellence, and commitment to education, Dr. Ragini Singh continues to make meaningful contributions to biomedical science and healthcare innovation in India. I would now request Professor Ragini to continue her presentation. Thank you, ma'am, for joining us today. It is an honor and pleasure to welcome you. Thank you, ma'am. Over to you, ma'am. We are able to see your presentation, ma'am. Yes. Full screen as well, ma'am. Thank you. Over to you. Please switch on your mic, ma'am. Mic is off.
Yeah, I hope I'm audible now.
Yes, ma'am. Yes.
Okay, thank you so much.
Thank you.
Good morning, everyone. Thank you so much, Dr. Vijay, for the kind introduction. At the start, I'm grateful to the organizers and especially Dr. Vijay Chaudhary for inviting me to speak today and for the opportunity to share the insights on emerging biomarkers in advanced lipid testing. So after the lucid presentation by Dr. Madhusudanan, UK, on genomics and proteomic biomarkers in rare neurodegenerative disease, let's shift our gear to more common disease, which is cardiovascular diseases. The title of my talk is Advanced Lipid Profiling with the Emerging Biomarkers, a paradigm shift in cardiovascular risk assessment. Now, the title has been chosen with the purpose that we need to answer one question. Why are we talking about lipids today? For most people, the word cholesterol itself brings some fear of some fried foods, ghee, or butter. But as biotechnologists, you know that lipids are much more than a dietary villain. So they are fundamental biomolecules and they come in many forms like triglycerides, cholesterols, lipoproteins, forming cell membranes, storing energy, and acting as precursors for various hormones like cortisol, estrogen, testosterone, vitamin D. Without lipids, no cell would survive. Yet, the same molecule that builds life can also threaten life when they are mismanaged in our blood. So let's take a quick example that not only introduces us to the basic concept of blood lipids, but also their significance. So imagine your blood as a busy highway. So lipids are the cargo, and the cargo, that is triglycerides and cholesterol, it is to be managed by a principal organ, that is the liver. But I hope you all remember that lipids do not float freely in water. So they need certain trucks, and these trucks are your proteins termed as apoproteins to transport them. Now these trucks, they are termed as lipoproteins, a complex of lipids and proteins. So some of them, they are the safe transporters termed as high density lipoprotein, abbreviated as HDL, while others, some of them, they are termed as rash drivers that can cause accidents. That is the low density lipoproteins, abbreviated as LDL. And they can cause accidents. And these accidents are what we call atherosclerosis or clogged arteries that can eventually lead to cardiovascular disease. So understanding lipoproteins is like knowing which drivers, that is the HDL or the LDL on the road, that is in the blood, are disciplined and which are more dangerous. So when we talk about HDL and LDL, the good one, the better one, the safe one is your HDL, that is the high density lipoprotein. And the dangerous ones are your LDL, subcategorized as large LDL particles and small dense LDL particles. So the figure which is shown in the slide, you can appreciate that the liver is serving as an organ which will be distributing the cargo, that is the lipids, the triglycerides and cholesterol throughout the body. And it also serves as an organ which will take back the cholesterol which is there in the peripheral tissues back to the liver, means it will be conveyed back to the liver where it will help in its excretion. So when we talk about liver, the liver will release the cargo, that is triglyceride and cholesterol, in form of lipoprotein particles, that is VLDL, that is very dense lipoprotein particles, which eventually when they travel through the blood, they donate their cargo to the tissues, to the tissues, peripheral tissues, and they gradually gets converted to intermediate density lipoproteins and eventually to LDL. If you can appreciate from this figure, we have two varieties of LDL here, which is a new coming concept. One is your large buoyant LDL, which is containing higher concentration of cholesterol in comparison to small dense LDL, which is containing lesser amount of cholesterol. So of all these lipids, so if you see the cargo which is released by the liver, the triglycerides and cholesterol released by the liver in the bloodstream, these lipoproteins are basically atherogenic in nature, whereas the lipoprotein, which is HDL, it is termed as anti-atherogenic. Why? Because it is bringing the cholesterol from the peripheral tissues back to the liver for its elimination. Now, when we talk about routine blood lipid panel, I'm sure many of you must have seen certain clinical reports. Some of your family members, they may have been advised by the clinicians to undergo a blood lipid panel. So what are the fractions which are being represented in a blood lipid panel, usually commonly by the laboratories? So there are basically four key lipid fractions which are generally seen in the reports given by majority of the laboratories nowadays. That is total cholesterol, which is actually the sum of all the cholesterol in the blood, but it is a very crude measure. Then there will be a report on LDL cholesterol, termed as LDL-C, so which is the so-called bad cholesterol, and many a times the scientists also refer this as a lousy cholesterol, and it carries the cholesterol from the liver to the peripheral tissues. And then we have the good cholesterol, that is the HDL cholesterol, which is helping in reversing the cholesterol which is there in the peripheral tissues back to the liver. And then there are levels of triglyceride, which is the major form of fat storage in the body, in the adipose tissue. And remember, higher levels of triglycerides is linked with insulin resistance and metabolic syndrome. So metabolic syndrome is a composite term given for 5 main factors, which is one of which is high triglyceride levels, high blood pressure or hypertension, low HDL, and high blood sugar, and definitely the central obesity. Now, on the left side of the slide, on the right side of the slide, again, I have shown a very simplified version of the lipid molecules, how they are managed by the liver and the heart and the peripheral tissue. So here from the liver, the cargo is released as VLDL. Gradually, it is converted to LDL because it donates its triglyceride and cholesterol to the peripheral tissues. And this LDL, it can deposit cholesterol to the heart and peripheral tissues as well. And from the peripheral tissues, cholesterol is taken back by HDL and brought back to the liver. So in a nutshell, from this slide, it says that LDL is bad cholesterol and HDL is our good cholesterol. So these fractions, they actually look too simplistic, right? But there is a problem in this. What is the problem? So let's try. This was the basic lipid profile, which you must have seen in many of the medical reports. Now let's move from basic to advanced. So this slide is little difficult to comprehend, but I would like to walk you through in a simple way. So the section, above section shows various types of lipoproteins which are present in the blood. So lipoproteins, as I mentioned, they are complexes of lipids and proteins, right? So they are spherical molecules. So on this side, we have the chylomicron having one protein component, which is ApoB48, which is converted to chylomicron remnant. So where are these fractions formed? This fraction is formed from the dietary lipids which you are taking, right? So they are released in the intestine and they are formed there. And gradually on the other side of the panel, you will see other lipoproteins, which is VLDL, intermediate density lipoproteins, low density lipoproteins, and LPA. Now, what is the common feature among these lipoproteins? Number one, they are all, the VLDL is released from the liver, gradually gets converted into these molecules. Additionally, they also have one protein component, which is termed as ApoB100. All of them, if you see, they're having this ApoB100 associated with them. Now, when I was talking about the basic lipid panel, I did talk about VLDL, LDL, triglyceride, HDL, but I did not mention anything about lipoprotein with a little a. Now, this molecule is an independent causal risk factor for cardiovascular disease. It differs from LDL in a simple component, which is there is one presence of one more protein. In addition, means one protein is added to LDL and it gets converted to a molecule which is called as lipoprotein little a. I will be talking more about these molecules during the progress of my lecture. But what I want to say is that the chylomicron and chylomicron remnants, the dietary lipids which you have consumed, they stay in the blood for refractory time. Whereas when I talk about the other lipoproteins, which is VLDL, ILDL, LDL, and lipoprotein A, they stay in the blood for the longer time. Now, the question here is, why are we talking about the lipids today, and why are we talking its association with cardiovascular disease? So to give you an example, what we will be discussing over next 15-20 minutes, I would like to give you one very crucial example here. So you may encounter two people having the same LDL cholesterol. So I think by now you can appreciate that LDL cholesterol is a bad cholesterol, which clinician will every time want to see the report of when you report for some when you are being diagnosed for diabetes and all. So he would want to assess the LDL cholesterol level. And we have these two individuals who have got the same LDL cholesterol value, but do you say they will have the same risk towards cardiovascular disease? The answer is no. One may have larger LDL particles in the blood plasma and the other may have small dense LDL particles in the plasma. So what difference does that bring? The larger LDL cholesterol particles, they are more buoyant in nature, they contain more cholesterol. Whereas the smaller dense LDL particles, they are in a way can be easily oxidized. They have lesser amount of cholesterol and they are the ones which are causing more damage to the endothelium and eventually causing cardiovascular disease risk. So what we are saying, why do we say that there is a paradigm shift in cardiovascular risk factor assessment? So till now, what we were looking for simple fractions in the simple blood lipid fractions, that is total cholesterol, LDL cholesterol, HDL cholesterol, and triglyceride. But now what we are coming up to is the advanced lipid markers, where we are trying to assess the type, the nature, the particle number of these fractions which are there in the blood, which can give a more composite idea about the individual's risk towards the cardiovascular disease. Now, why it is relevant today? Why are we talking about moving from basic lipid profiling to advanced lipid profiling? Why this is relevant? This is relevant because when we look at the statistics, so cardiovascular disease is one of the number one cause for death in India, and Indians develop this heart disease almost 10 years earlier than the Western population. So that is the reason we are trying to look towards advanced lipid profiling. Now, unlike the West, where obesity-driven high LDL is the main culprit, in Indians, we often see low HDL, high triglycerides, and normal LDL, which is a hidden risk profile. Now, identifying specific risk factors for cardiovascular disease not only enables clinicians to treat the patient, but also personalize the therapy for the aberrant lipid profile and other cardiovascular condition according to each patient's individual risk profile. Now, this type of approach is called as the personalized approach, and it not only maximizes the therapeutic effectiveness, but also improves the clinical outcomes. Now, this risk stratification has become the foundation of modern preventive cardiology, which is having solid base in advanced lipid profiling, allowing for targeted interventions based on comprehensive biomarker assessment rather than isolated laboratory values, which is the usual practice in India still. Having said that, I would like to highlight the major coronary artery disease risk factors in the Indian population. So in India, the traditional risk factors which are associated with cardiovascular disease risk is definitely the dyslipidemia, then smoking and hypertension, but metabolic risk factors, they also dominate. Central obesity, that is the pot belly, is more predictive than BMI. Then family history, in Indians, it is strong. Indians often carry alleles for lipid abnormalities. Then we have PCOS, polycystic ovarian syndrome, a growing problem in young women, increasing their metabolic risk. So I would like to say that if cardiovascular disease is a fire, in Western countries, the main fuel is LDL cholesterol. And when we talk about the Indian context, the fuel is more like a mix of low LDL, high triglyceride, and insulin resistance. So that brings me to the idea of atherogenic dyslipidemia triad, which is more common for the Indian population. So that is number one, elevated triglyceride values. So increased triglyceride levels often reflect what? It reflects insulin resistance and excessive VLDL production. Then we have small dense LDL particles. So I have already mentioned about the differences between large buoyant LDL particles and small dense LDL particles. So among the two, these are the culprits. So increased blood concentration of small dense LDL particles that are more susceptible to oxidation and more atherogenic than larger, more buoyant particle. So if an individual has higher concentration of small dense LDL particle, his risk towards cardiovascular disease will be high in comparison to individual who has got similar LDL values but has got more buoyant LDL particles in the blood. Then definitely the decreased HDL, reduced HDL level particles not only reduces the reverse cholesterol transport, which we have already discussed, but also it diminishes the anti-inflammatory protection, which is given by the HDL. So I would like to say that you can imagine the LDL particles as cricket balls. So small dense LDL particles, they will be like tennis balls in a wet pitch, and it can move unpredictably and damage the vascular wall more easily. Whereas the large buoyant particles, LDL particles, they are more like a proper leather ball, which are less dangerous, and Indians unfortunately carry more of the small dense LDL troublemakers. So understanding the type of LDL particles is more significant than just knowing the LDL cholesterol values. So that brings me to understanding the causes for this aberrant lipid profile, which is seen in the Indians. So we do see cases with increased total cholesterol and increased LDL cholesterol. So these things are commonly observed in India, and the reasons being hypothyroidism, especially in women, who often shows up first as high cholesterol, high alcohol intake, rising among the young men, which is a concern, is also a strong cause of high triglycerides, and definitely the overuse of steroids or hormonal treatments in infertility cases may also contribute to increase the risk of an individual towards cardiovascular disease. More specifically, I would like to emphasize here, particularly the gym regulars. So for students, it is I believe in this age, the the audience here, I believe many of you must be regular goers for the gym. Just be very careful and watchful what steroids, anabolic steroids, your gym trainer is prescribing to you, because that may cause dyslipidemia and eventually may increase the risk towards cardiovascular disease. So, so remember, we do also have a profile where we have increased triglycerides, increased very low density lipoprotein levels. Again, the reasons can be metabolic, which I've already talked about, like type 2 diabetes, obesity, excessive alcohol intake, hypothyroidism, certain medications, and definitely the steroids, anabolic steroids, which I have talked about. So remember that these secondary causes are not just the textbook facts, they are everyday realities in the Indian clinics. Having said that, we now move on to the cholesterol controversy here. So this cholesterol controversy is, it actually centers on the premise that cholesterol is the major component of atherogenic fatty plaque. That is what we are all, the print media and the articles which are not based on sound evidence, they have made us believe that cholesterol is the culprit for causing atherogenic plaques and eventually the cardiovascular disease. However, this view is very oversimplified version of a complex pathophysiological process like atherosclerosis. Now, what is clinically important in atherosclerosis more than cholesterol is the distribution of lipoproteins and their respective subfractions. As I've just mentioned, LDL subfraction, the buoyant one, and the small dense LDL, as well as measurement of other components of the lipoproteins, which are being added in advanced lipid profiling, the apolipoproteins, the ApoB, which I have already talked about, the ApoA1 associated with HDL. They provide a more nuanced insight into the cardiovascular disease risk. Now, modern risk assessment not only requires us to look beyond the traditional lipid fractions, which I mentioned at the start of my lecture, but we now also want to look at the particle composition, the lipid particle composition, that is the HDL subfractions, their numbers, their size, and their quality and their functionality as well. So cholesterol definitely, after this cholesterol controversy, I would like to say that cholesterol definitely is not the villain here. It is essential to make hormones like estrogen, testosterone, cortisol, and vitamin D. So the problem here is when it is in the wrong package. So HDL is like your Swachh Bharat mission truck, cleaning up excess cholesterol from the peripheral tissues, bringing it to the liver, whereas low density lipoprotein, that is the LDL, is like a careless dumper truck, which is dropping cholesterol garbage in blood vessels and causing atherosclerotic plaque and eventually cardiovascular disease. Now, I would like to give you example of importance of LDL subfraction with this slide. So you can just imagine an example of two Indians having an LDL cholesterol value of 120 milligram per deciliter. So one has in his blood the larger LDL buoyant particles, whereas the other one has small dense LDL particle. So I think now you can appreciate that this particle heterogeneity is the cause of different cardiovascular disease risk in the two individuals. So the individual who's having small dense LDL particle will have higher risk towards cardiovascular disease in comparison to the individual who's having large buoyant LDL particles. So the point here is that simply estimating LDL cholesterol will not help in assessing the hidden risk. So this explains why many Indians who are slim, vegetarian, and non-smokers still develop heart attacks at a young age, because standard LDL testing, the traditional LDL testing misses the small dense LDL problem. That brings me to one more concept here, which is non-HDL cholesterol. So non-HDL cholesterol, so this figure, if you appreciate here, I have shown the different lipoprotein fractions. Chylomicrons are the lipid fractions which are coming from the diet. Rest are synthesized by the liver, and some are formed during the course when VLDL moves in the blood. So of all these, on the left side, apart from HDL, they are your atherogenic lipoprotein particles which constitute the non-HDL cholesterol. So this non-HDL cholesterol, which is total cholesterol minus HDL cholesterol, provides a more comprehensive assessment of atherogenic risk than LDL cholesterol alone. Why I'm talking about non-HDL cholesterol here? Because many clinicians at times when you visit them, they would only want to give you, they would only want to advise you to undergo LDL cholesterol testing. They would not even want to look at the other lipid fractions, which eventually can give you a kind of peace that your LDL cholesterol is in the established biological reference interval, but eventually he has not calculated any of your risk associated with the other lipid fractions present in the blood. So non-HDL cholesterol actually captures all the atherogenic lipoproteins, including LDL, VLDL, IDL, and the lipoprotein A, and is more closely related with cardiovascular disease risk than LDL cholesterol alone. And it is particularly important when the triglyceride levels exceed some 200 milligram per deciliter. And it provides a more accurate risk in metabolic syndrome and diabetes, and it requires no additional testing. So the same blood sample which you have given for LDL testing, that on the same sample, the other markers can also be analyzed. So it is actually giving you a complete picture of all the bad buses which are present in the plasma on the arterial highway. So it is not just the LDL buses, you can have other bad buses as well. So this brings me to the limitations of the standard lipid testing, also known as traditional lipids testing. So traditional lipid testing, as I said, it is composed of only total cholesterol, LDL cholesterol, HDL cholesterol, and triglyceride. It does not give you a complete picture of your cardiovascular risk. So the normal LDL paradox I have already talked about. I gave you an example of two individuals having the same LDL values. So why this discordance? The discordance is because we are not estimating the actual particle number, and we are not estimating the apoproteins which are there with the atherogenic particles. So that brings me now to the advanced lipid profiling. So this advanced lipid profiling, what it is actually. So this advanced lipid profiling will go beyond the traditional 4 measurements which I have been talking about. It will give you more detailed insight into the composition, the number, the size, and density of lipoprotein particles that drive atherosclerosis. So the comprehensive particle analysis is important apart from measuring the routine lipid fractions. So this comprehensive particle analysis will include your measuring the LDL particles, their number, HDL particles, their size, apoproteins associated with these lipids, that is ApoB and ApoA, and most importantly, the newer marker in the horizon, which is your lipoprotein with a little a. So the analytical methods used to analyze these is definitely NMR, which is actually costly as per the Indian scenario. So I would like to talk a little bit about lipoprotein little a. So this lipoprotein little a is often called a South Asian curse. So if you can appreciate in this figure, the structure of LPA, lipoprotein with a little a, is somewhat similar to LDL, but with an extra protein attached, which is apoprotein little a. Now this extra protein which is attached here, it is acting as a Velcro tape, and it will go and bind to the endothelium and cause damage and can lead to atherosclerosis. So this elevated LPA, it promotes clotting as well as plaque formation. Ironically, this elevated LPA is resistant to both the lifestyle modifications and medications, and is a genetically determined factor whose values get fixed at around the age of 5. So if we say that LDL is like a careless bus driver, so LPA is that same bus but with carrying a load of explosives with it. So it is genetically determined. So lifestyle does not change it. So that's why family screening is vital here. And many guidelines, many international guidelines now recommend testing of LPA once in a lifetime for screening in all the population to reduce the risk towards cardiovascular disease. So I've talked about ApoB, estimation of ApoB, and I've talked about the presence of ApoB on the atherogenic lipid particles. I've talked about presence of ApoA1 on HDL particles. But what I want to mention here is that in India, many people, they will have normal LDL, but they will have high ApoB. So that is why estimating ApoB becomes crucial, because it will give you a clear picture of the number of atherogenic lipid particles which are there in the blood. Then I will talk a little bit on HDL subfractions also, because I've been talking more on LDL subfraction. HDL subfractions are also there, but HDL2 and HDL3, so HDL2 versus HDL3, more of the function of HDL for any cardiovascular risk assessment, the functionality of HDL is critical. So the large HDL subclass particle, that is the HDL2, is closely associated with cardioprotective function. However, recent research indicates that the medium-sized particles, that is HDL3, may offer protection against coronary artery disease and cardiovascular disease. Now, HDL not only exerts anti-atherogenic properties, it has also got anti-inflammatory actions inherent because of presence of certain enzymes like peroxygenase. So LDL types and particles I have talked about. So greater number of LDL particles, the risk becomes high. Greater number of small dense LDL particles, the risk multiplies on that. Now, apart from this, so we have been talking about one spectrum which is actually causing the cardiovascular disease, that is dyslipidemia. On the other end of the spectrum is the inflammation. So the advanced lipid profiling will also include certain markers which are related to subtle inflammation, which is present in individuals who suffer from diabetes mellitus, right from the time of diagnosis. And these markers are your HSCRP, which is high sensitivity C-reactive proteins, then lipoprotein associated phospholipase A2, then fibrinogen, homocysteine, and surprisingly vitamin D is also there. Then definitely insulin, certain stress markers like NT-proBNP, then TMAO and omega-3 and omega-6 ratio, and genetic testing. So I'll talk a little bit about LPPLA here. This is an enzyme which is actually produced by the intima of the capillaries, and it is based and it is released when there is atherosclerotic plaque development, and it is a vascular specific inflammatory marker. Homocysteine, it can exert a wide variety of effects expected to promote thrombosis and cardiovascular disease. Basically, it alters the expression of adhesion molecules, cytokines, which play a critical role in generation of atherosclerotic plaque. And then fibrinogen, all know that it is a principal clotting factor. So it has got key role in arterial occlusion by promoting thrombus formation, endothelial injury, and hyperviscosity. So these are also the markers which are suggested to be estimated during the course. So homocysteine, I would like to say that Indians definitely, because we have common folate and vitamin B12 deficiency, so because of the vegetarian diet, so that brings us in a zone of having elevated homocysteine level and thereby increasing our risk towards cardiovascular disease. And when I talk about vitamin D, how it is associated, so vitamin D, despite abundant sunlight, more than 70% of Indians, they are deficient due to lifestyle and skin pigmentation. And deficiency definitely worsens the vascular calcification, which is happening at the atherosclerotic plaque. So vitamin D also, estimation of vitamin D also plays a crucial role here. So the other molecule which I would like to talk is trimethylamine henoxide, looking at the younger audience here, because the younger audience, they are in habit of consuming more of eggs and then the energy drinks. So when you consume more of these things, that leads to generation of trimethylamine henoxide, which actually upregulates macrophage scavenger receptor actions, and more of cholesterol is captured, and that can lead to formation of the atherosclerotic plaque. So cardiovascular biomarker panel starts from the basic markers. So four of them we have talked. Additionally, depending upon your condition and when you present to the clinician and the available laboratory facilities, I would suggest that these markers should also be added to assess your long-term risk towards cardiovascular disease. So expanded lipid profile, it measures the lipids The lipids are fractions, their size, the numbers, as well as certain apoproteins like ApoB, ApoA. So this profile should be preferred over the traditional lipid profile if you are really really wanting to assess your risk towards cardiovascular disease. Definitely, the expanded lipid profile requires more advanced technologies like NMR, which is little difficult at this stage in India, but I'm sure with inputs from students from Indian biotech innovation, it will soon become more affordable. So this is where I would want to say that biotechnology students should develop cost-effective indigenous assays so that millions of Indians can benefit without spending thousands on foreign kits. So in India, such panels can help us identify young, seemingly healthy individuals at risk, like software engineers with sedentary jobs or women with PCOS who look slim but carry metabolic disturbances. So the take-home message here, I would like to say that Indians, they definitely have a unique lipid phenotype that is low HDL, high triglyceride, high small dense LDL particles, high lipoprotein with a little a. Traditional cholesterol test, traditional lipid panels, they miss these tests and definitely underestimate your risk towards cardiovascular disease. Advanced biomarkers will provide the missing link for early detection. And for biotechnology students, this is definitely an opportunity to develop low-cost, reliable assays tailored for Indian population. And so on the closing, I would like to say, so the next time when you think of cholesterol, don't just imagine butter and ghee. Think of complex molecules, genetic predispositions, gut bacteria, and inflammation. And remember, if biotechnologists can make affordable assays for these biomarkers in India, you won't be doing just science, you will be saving millions of lives. So that is all from my side. I hope I could justify my presentation, giving you an insight about comparison of traditional lipid profile versus advanced lipid profile, the importance.
Yes, ma'am. Thank you so much. It was such a wonderful presentation, and it actually gave insights into how we can prevent cardiovascular diseases. In fact, all of us are into the habit of eating a lot of pizza, burger, and all, which is our favorite foods over the time. So even young generation, for them, they if they think of a party, they think of pizza or burger, isn't it? So I think that's the way we should actually think when we think about our intelligent choices of food. When we think about parties also, because cardiovascular disease is on the rise, as you have mentioned rightly, and it is not only the lipid profile which has contributed to the cardiovascular disease, as you have rightly mentioned. So traditionally, we only go for checking LDL or VLDL and HDL profiles. We don't even total cholesterol. We don't even look into the advanced diagnostic protocols which are available.
Right.
And I think this must have been an eye opener to all the audience who are sitting here, especially to make intelligent choices in their food. And of course, very heavy exercises are also not preferable. So
Yeah.
So you need to have a balance in your life in terms of your food, the risk which you take in terms of stress, and more importantly, vitamin D as well as inflammation. Their contribution is also very important. And
Yes.
also the hormonal imbalances like hypothyroidism, which you have mentioned. So it is
Right.
really a wonderful, insight and informative lecture to all of our students. In fact, I would like to share with you, unfortunately, we lost one of our students who was a a master student in business studies in our university. And she did not wake up in the morning one day in her PG. And we came to know that she had a cardiac arrest in the night. So it has become so widespread even among the youngsters. So that is really alarming. So I think this has been such a wonderful informative session for all of our students. So let me
Thank you
just
so much.
ask a few questions from my students if they have any.
Yeah, I would be happy to answer.
Yes, ma'am. Anyone? Anyone? Any question? No questions? If you have any, you have the right time to ask right now. Don't be shy. Please come forward and ask. Yeah, it's still, ma'am.
I would be happy taking questions over my mail ID also, which you can find on my website, Ames Rajput website. So I'll
Yes,
be
ma'am.
happy.
One student is asking question.
Okay,
Just one
okay.
second, ma'am. Thank
Yeah.
you. Come, please.
It's about cancers.
Yeah, yeah, ask that.
Yeah. My question is, what role can R and AI play in cancer therapy?
RNAi therapy in cancer. See, RNA interference. Definitely, this particular technology, this comes under gene editing. So we can definitely identify markers related, means the RNA-based markers, which are showing aberrant expression in tumor tissues, tumor cells, and then we can, making use of this particular technology, we can silence their expression. So that is one way how we can make use of this technology.
Yes, ma'am. Thank you so much for answering question. Now, one more question is there from the audience, ma'am. Our
Yeah,
faculty,
please, I would be
Dr.
happy to.
Priyanka ma'am is asking.
Yeah, please.
Hello, Dr.
Hello.
Raj, this was a very nice and informative. And actually, I could read it because my sister was diagnosed with like a high risk of CVD, but we were
High
not
risk of?
able to understand because the lipid profile, like the different parameters, HDL, VDL, they all got okay, but still the blood pressure was very high. So I think now I'm able to correlate why advanced diagnosis is really needed. So thank you so much for this information. My question is like, is there advanced diagnosis is available in the market or like this is a future perspective only?
Few of the markers, certainly, ma'am, they are available in the market. Say, for example, LPA with little a, then inflammatory markers, they are available in the market as of now. But for LDL subfractionation and HDL subfractionation, we are still a country where we have not started this in routine clinical practice. But yes, certain indices, if you calculate those indices based on your HDL and LDL fractions and cholesterol fractions, they can also give you an idea how you can have an means indirect correlation towards what do I say, the fractions, the buoyant LDL fraction and the other small dense LDL fraction. So if you would want, you can, if you want, you can share the report with me and I can help you with that. Probably Dr. Vijay can help us to communicate regarding this. So that I can do. But some measures, definitely the inflammatory markers, which I said HSCRP is being routinely done even in our institute. We have also initiated LPA measurement at our institute because we understand that this is a genetic risk marker. And as I mentioned in my talk also, every individual, specifically we as South Asians, we have higher risk towards coronary vascular disease. We should get that estimated. So that is what I can say as of now.
So since this NMR, like lipoprotein is a future perspective only. This is not
In our country, yes, it is a future perspective. In European nations, they have already started advanced lipid profiling with NMR. They have started.
okay. Thank you, ma'am.
Thank you, ma'am. I think even homocysteine marker can be done, right, ma'am?
Yes. Homocysteine can also be done. Yeah.
And that is available.
Homocysteine, yeah, that is available.
Yes, ma'am. Any other question from the audience? Okay, ma'am. Thank you. Thank you so much for your time, ma'am, and thank you for joining us today and giving such an informative lecture. It was really wonderful to listen to you today morning. Thank you for joining us,
Thank
ma'am.
you. Thank you so much. Thank you so much.
Thank you, ma'am. Now we move on to the oral presentations and poster presentations. A few are left today, so we move on to that. And for the oral presentations, I would like to invite the session chair, Dr. Priyanka Singh. Dr. Priyanka Singh is currently working as Associate Professor and Head of the Department at National Institute, NIMS Institute of Medical Science and Technology. She has received DST Inspire Fellowship for her PhD work and has established herself as independent investigator after joining as Assistant Professor at Banasthali University. She has successfully contributed her academic excellence to enhance reputation of allied medical science and technology as a best teacher and research department. Then, driven by a passion of science and education, she wants to settle herself as women entrepreneur dedicated to nurturing women scientists. Her research area is mainly focused on bioprocess technology, nanotechnology, biofuel production, enzyme technology. She has published more than 50 research papers, 10 book chapters, 2 patent filed, 2 books in reputed SEI High Impact Journal and International Publication House. As leading women scientist, she has received Outstanding Researcher VIHA Award, Eminent Scientist Award, Best Innovation Award, Best Researcher Award, Indian Education Award for her research excellence. Her vision is to scale up technology for production of value-added industrially significant products to contribute sustainable goal of country and create employment opportunities for women. Thank you so much, ma'am, for joining us today. Over to you, ma'am, for the opening remarks.
Yeah, thank you, Dr. Viji, to give us such a wonderful introduction for me. And really, I am very much thankful to all the organizing committee member for this conferences. They have invited me for the session chair here. And but I think in this oral session, a student will be also benefited with their reviews and questions, query for their improvement of their research paper. So, Dr. Vijay, we can go ahead for the starting of the session.
Yes, ma'am. Yes. So I would like to invite Nanam Ronia to kindly start your presentation.
Hello,
Yes.
am I
Yes, please.
audible?
And yes, you are audible. Please share your presentation.
Okay. Hello, I hope it's visible.
It has not come yet. Is there an issue from your end? Yeah, it has come up now. Yes, please.
Okay, should I start, ma'am?
Yeah, it is visible now. Kindly move ahead.
Okay. So a very good afternoon to all the respected dignitaries present here. My name is Miss Nanam Ronia, and I'm a PhD scholar in Department of Food Technology at Rajiv Gandhi University, Arunachal Pradesh. And today I'm here to give my presentation on my research work, development and evaluation of peanut milk-based vegan curd and kefir as dairy alternatives. So starting with the presentation, So starting with the presentations, as we all know, milk is the primary food for not only humans but all the mammals, as it provides all essential nutrients required for proper growth and development. But in recent time, due to the change in climates, growing demand and populations, the product of milk has hampered a lot. It has decreased, which which leads to the raising of dairy, the cost of dairy products. And along with that, there is also increase in demand of vegan food and lactose-free food. So this research aims to formulate peanut-based milk, curd, and kefir, which will be both nutritious and both affordable. So if we talk about background of the studies, as we have already discussed, there is rising concern and demand for lactose intolerance, milk allergies, and vegan dietary preference. So the question may be, why peanut only? Because there are varieties of dairy alternatives we have in the market, like almond milk, soya milk, coconut milk, etc. But as we all know, these all dairy alternatives are more expensive as compared to peanut, and peanut is readily available everywhere in low cost, and it is also a nutritious food. So the study designs to explore the nutritious part of peanut along with it, by producing probiotic products from it, that is curd and kefir. So in this study, firstly we have formulated the peanut milk by soaking the peanut, grinding it, and filtering it to formulate peanut milk. And after formulating, the peanut curd was prepared by boiling the obtained milk. After that, the curd culture was added for fermentation. And for the formulation of milk kefir, first the milk was heated and kefir starter culture was added, which was inoculated at 12 to 24 hours. So after the formulation, different evaluations was done. Firstly, the formulation was in 3 different forms. First, it was formulated with adding sugar, and then only culture and jaggery. And these 6 formulations, that is 3 of peanut milk curd and 3 of peanut milk kefir, was sensory evaluated using hedonic scale by trained professions. And among these 6 samples, the sample which contained jaggery, the jaggery was added in the samples, has found to have the highest acceptability rate in compared to the other samples. So with this result, we have chosen this jaggery curd and peanut kefir with jaggery for further analysis. And during the proximate analysis, it was found that although the carbohydrate level and ash content was lower in comparison to the normal dairy curd and kefir, but the moisture content, the fat content, and the acidity level was same as the dairy curd and kefir, and the protein content and crude fiber content was higher in comparison to the milk curd and milk kefir. So along with the proximate analysis, antioxidant activity was also studied, where the anthocyanin level, flavonoid level, and antioxidant level was studied, in which the anthocyanin level and the DPH level was found to be in low to moderate range, but then the flavonoid content was found to be higher than the normal milk curd and kefir. And this flavonoid content could be due to because we have added the peanut skin also. So with this result, the microbial analysis was also done, and microbial load was found to be higher in comparison to the commercial curd and kefir. So with this, I would like to conclude that the peanut-based milk peanut-based curd and kefir has shown has shown potential as nutritious and functional food. Though the jaggery variant was most favorable among the sensory evaluators, and it has found to have notable flavonoid and microbial activities. So these are the references, and thank you everyone.
Yeah, Nona,
Yes, ma'am.
you have told कि you have taken the peanut milk and you have synthesized the curd and kefir. Okay.
Yes.
Okay. For the curd synthesis formulation, you have done the fermentation process also. So can you tell me which type of microbes you have taken for this fermentation process?
Ma'am, we have not taken specific microbe. We have taken the curd culture that.
So what is the culture? What the specific culture you have taken?
Hello, Ronia, is it
Yeah.
audible? Ronia?
Hello?
Yes, Ronia, please go ahead.
Okay, sorry, actually the network.
Without the adding of any culture, can it possible that milk will be converted to the curd?
No, ma'am.
No. And second thing, in the progumate analysis, can you show me that's again that histogram, that chart to me? That progumate analysis you have shown for the jaggery and for the curd.
Okay. Ma'am,
Ronia,
regarding the first
that
question.
chart, no,
Yes,
that chart you have shown in your resultant analysis for the progumate analysis. Please show me that chart.
yes, ma'am.
No, previous one. Yeah, this one. Okay. In the peanut milk curd and the kefir, okay, you are going to observe the same amount for this moisture and that CHO. What is this CHO?
Ma'am, carbohydrate.
This is the carbohydrate.
Yes, ma'am.
Which type of carbohydrate you have analyzed here? Is it lactose only?
Yes, ma'am.
Okay. Then you have told me that milk curd is मतलब कि according to this progumate analysis, this milk curd is highly effective in compared to the milk kefir. Only on the basis of these two factors, moisture, high level of moisture, and this high level of the carbohydrate. That is not the higher level also. 82.7 is your moisture content, and this is only for the 88.7. Okay, Ronia, one more thing. You also tested the antioxidant activity also, no? Ronia, am I audible?
Hello?
Antioxidant study
Ma'am,
also
can you
you have
please?
shown, no? Antioxidant study also
Yes.
you have shown, no? Can you tell me what are the methods
Yes,
you
ma'am.
have applied for this antioxidant study?
Ronia, can you hear us? Ronia?
Ronia, am I audible now?
I think there is some internet issues, ma'am, I think.
Yeah, it's okay, Dr. Vijay, है ना?
Okay, ma'am. Okay. I think we can take the next oral presentation candidate. Ms. Simran Singh. I think she is not available. Ms. Pooja Barua. Ms. Pooja Barua, are you here? I don't see her as well. Ms. Varsha R.
Yes, ma'am.
Okay, Ms. Varsha, please, start your presentation. Naniam, can you please unshare so that Varsha can share? Nana Bronia, kindly unshare so that Varsha can present. Yes, Varsha, please go ahead. Your voice is breaking, Varsha, and screen sharing has not started yet.
Visible?
It's not visible yet. Yeah, it has come up now. Please make it full screen.
Am I visible? I mean, can you
Yes.
hear me?
Yes. If you can switch on your video as well, then it will be good.
Okay. Shall I start with my presentation?
Yeah, can you please switch on your video as well? Hello, Varsha. Yes.
Hello, am I visible, ma'am?
Yes, you are visible and you are
Good
audible.
afternoon, everybody.
Go ahead.
Okay, thank you. Good afternoon, everybody. Today I am going to present my paper. I am doing my MSc in biotechnology from Ramaya University of Applied Sciences. This is my collaborative work with the HOD of the department, Dr. Shruti. It is a comparative analysis of bacteriophage present in Ganga water sample collected before and during the Kumbh Mela. Uh, Kumbh Mela, we all might have heard about the massive spiritual gathering which happened in Prayagraj in 2025. A massive influx of 57 crore people had attended this holy spiritual gathering. During this gathering, there were a lot of spiritual activities happening along with devotees taking holy dip into the water. One would believe when such influx of people taking holy dip in the water, there will be an increase in bacterial infections, and people might have got affected from the disease, from the dipping as well. These are some news articles stating that UP government states that there was no reports that people got affected from this holy dips, and it was unaffected despite the holy dip. You can see the articles which is present on the screen at the moment. So my study offers the potential as one-stop problem to two problems, that is a pathogenic load in water, which helps us assess the pathogenic load in water, and also as an environmental monitoring of the water quality, also using bacteriophages. We all know bacteriophages are virus which particularly attacks a particular bacteria. So we chose bacteriophages over bacteria because bacteriophages directly don't affect the human, but bacteria do. The methodology includes when we took two types of bacteria. One is Escherichia coli and one is Staphylococcus aureus. Both are major indicators of contamination, to which we grew the bacteria in LB broth and inoculated them with the bacteria. And then we did an double layer agar overlay and created a lawn of bacteria onto the Petri plates. After which 50 microliter of the water sample collected before the Kumbh Mela and after the Kumbh Mela was added into the sample. Talking about the sample collection, many people from my institute and my professors had been visiting the site personally. So the water collected from them was by them, but the exact location of the water sample collection can't be located. So these are my results. You can see E. coli sample before comb and during comb. It is evidently clear that during comb the bacteriophages have increased. It shows a clearing zone. The lawn has been cleared into the circle. Clearing zones are seen. We have also taken as tap water as control, tap water and distilled water as controls to compare whether the bacteriophages are present in these water samples as well. The same thing was repeated with Escherichia coli before comb and during comb. In the during comb sample of Escherichia coli, you can see the bacteriophages load is so high that you can't even see the lawn of bacteria compared to that of the before comb samples. This is the tap water samples of the same for Escherichia, Staphylococcus aureus. This is a table showing my calculation for the plaque farming unit, which was calculated by number of plaque counted into the dilution factor by volume plated. So the dilution factor which we took was 10 to the power minus 9. And when you see the comparison, we can see that during comb, the plaque count is significantly higher compared to that of before comb. We have made 3 replicates of the same, and we have taken the average, and it shows the same results as that during comb the bacteriophage load is increased. Significance of this study is that environmental monitoring tool, it is a major environmental monitoring tool, because previously nobody has been monitoring the water quality by bacteriophages, but by the load of bacteria. Also understanding the microbial shift, because there's a load of people taking holy dips in the water, and public health implication by infectious disease which might be caused. Limitation, as I mentioned before, it was previously the geographical origin of the water was not known, because we collected it ourselves, and validation of these water samples can't be precisely located. These are my references. Thank you so much for your attention.
Yeah, Varsha.
Yes.
Am I audible, Varsha?
Yes, ma'am.
Okay, okay. You have told ki the water you have collected, from there you have isolated the strain E. coli and Staphylococcus. 2 strain you have isolated. How you have identified ki this is the E. coli and this is the Staphylococcus aureus?
Ma'am, actually, I took a bacterial count initially before starting the experiment. From that, I isolated the bacteria which I've seen in the plates, and done a gram staining to identify that there is a load of E. coli and Staphylococcus aureus in higher limits. So I took those samples as my major component for
From
the
the staining method, though, you can only identify now this is a gram positive or gram negative. But here you are telling about that the E. coli, that is the genus species, both you are just showing there. So I am just asking how you have confirmed that this is the E. coli strain. Have you done any sequencing analysis, any study?
No, ma'am, I have not done yet. I'm trying to do it after this session, because I'm pursuing to continue this project further with exact water samples from the exact locations.
Okay, for the bacteriophage analysis also, what are the zone of something clear zone you have observed? What is the size of that zone?
It was few zones were comparatively larger and compared to the other zones. One mm was mostly seen, the zone's diameter.
Okay.
Yes.
So what you have found ki before the 2 times water sample you have taken na, once before the comb and once after the comb. So after your analysis, what you are finally going to conclude? Which time we should visit the Kumbh Mela?
Yes, ma'am, what is the major understanding from my results is we would expect that during the Kumbh, the bacteriophage load is more. So we would think there is a large amount of bacteria in the water because there's a load of people taking holy dips. But because of the what I reckon is that UP government had taken a lot of preventive measures to take care of the water quality, and there were many from ISRO and BRAC where they took controls of the water. There were geo tubes which they used for cleaning out the water. So due to that, many infectious disease and bacterial load might have not affected the population at the moment. That was my conclusion.
Okay, okay, Basha, fine.
Thank you
Dr.
very
Viji?
much.
Hello, ma'am.
Yeah, ma'am.
Yeah, ma'am, I think the other two participants, they haven't joined. So,
Okay.
Vasha, thank you. I think we can conclude the session, ma'am. So I would like to have your concluding remarks, ma'am. Thank you.
Only the two presentation were there this time, Dr. Viji.
Yes, ma'am. I look forward to
It's okay, no problem. Yeah. And student also, whatever the shown the result that are also, I'm thinking is a very preliminary result they have shown here. Ma'am and Dr. Vinay Sharma, I especially thank you, sir. Ki, your team has organized such a very wonderful conferences, and you have invited us. This time, though, we have joined as a online. Next time, we will try to join in the offline mode.
Yes, ma'am. It
Okay.
will be
So,
more good and more happy for us to host
yeah,
you.
obviously, because I am to like a neighbor of your university, so we can also invite mother can do something with the collaboration also. But this time, this conference was really very good, and student has given a very good presentation here also.
Yes, ma'am. Ma'am, the next conference we are having in October, so we would like to have your students also joining us offline mode, ma'am.
Yeah,
You
obviously.
too, please, ma'am. Most welcome. We will be, requesting you also to join us, ma'am.
Yeah. Okay, ma'am. Thank you, ma'am.
Thank you. Thank you for your time, ma'am. Thank you so much, and
Yeah.
kindly share the results
Thank you,
of
ma'am.
the presentation.
Yeah, ma'am. Just now I will share within the 5 minutes. Okay, ma'am.
Okay, ma'am. Thank you. Thank you so much.
Okay, ma'am.
Thank you.
Good afternoon, everyone. Thank you so much for joining us today. And now we can all proceed for your lunch. And from 2 o'clock onwards, there is valedictory session at the auditorium. Okay, thank you so much. हाँ जी सर